Hello, everyone. Welcome to the trainee short communication session. We'll give another minute till about a quarter after the hour, and then we'll get started to let people join. Okay, I think in the interest of time, we'll get started. My name is Ken Hallows from the University of Southern California, Keck School of Medicine. And today, we will have 5 short presentations by trainees. Each one has an 8-minute slot, so about 5 minutes for presentation and 3 minutes reserved for questions. Our first speaker will be Navin Gupta, who has affiliated with the nephrology division at Mass General Hospital, as well as the Division of Renal Medicine at Brigham and Women's Hospital in Boston, and Harvard Stem Cell Institute. And he will be presenting a brief talk on human organoid-on-a-chip model for therapeutic development in ARPKD.
Hi, it's an honor to present in front of you today. My name is Navin Gupta, representing the Morazane Lab at Massachusetts General Hospital, presenting human organoid-on-a-chip model for therapeutic development in ARPKD. I'd like to preface to say that there are some, folks that are, have been doing this work for longer than myself. Those include Ken Hiratsuka, Tomo Miyoshi out of the Morazane lab here at MGH, and Katarina Kroll from Jennifer Lewis's lab at the Wyss Institute. And the work I'm presenting is in press at Science Advances. So we've been collaborating with Dr. Lewis for a number of years, combining our expertise in kidney organoids with her expertise in organ-on-a-chip techniques. And previously published in Nature Methods a few years ago that fluidic shear stress across kidney organoids induces maturation and vascularization. So here on the bottom left, you can sort of see a top-down view of such a chip. It involves a 3D printed PDMS gasket within which is a bed of proprietary extracellular matrix. Kidney organoids are placed in a central location and allowed to become partially embedded in this layer of ECM bed, and then subject to superfusate flow. And so the present work is really predicated on 2 presumptions. The first being that, polycystic kidney diseases are ciliopathies, and cilia transmit sort of mechano signaling, and that we thought that fluidic culture may induce cystogenesis in PKD organoids. The second premise is that kidney organoids are developmentally sort of mature enough to be able to model a developmental kidney disorder, ARPKD, caused by defects in the fibrocystin gene or PKHD1 locus in an autosomal recessive fashion. So in pilot experiments, we took PKHD1 double knockouts and then saw that when they were on chip and subject to flow, they developed cystic dilatation of CDH1 positive distal tubules, whereas in the absence of flow, there was no cystic dilatation. And so, after that pilot experiment, we were more interested in flow-derived potential, cystogenic signals. So the way we isolate what was flow-derived is via this schematic on the left. You can see at the top that we compared PKHD1 double mutants compared to heterozygotic mutant and did differential gene expression, looking for pathways that were really upregulated under, so we did these 2 conditions, the double knockout and the heterozygotic, in 3 different conditions under flow, Forskolin treatment, and static. And we wanted to understand the flow mediated signals that could be potentially novel cystogenic signals. So we took all the different, the pathways that were upregulated in the double mutant and negated the ones that were upregulated that could have been seen with Forskolin treatment or in a static environment to see these putative potential cystogenic signals. There are 353, did a literature search, 124 of them had been previously reported, 229 were potentially novel pathways. So we investigated certain specific targets, 5 from the 229 pathways that were sort of novel, unique ones that were putative cystogenic pathways under flow, and 1 target from a shared 32 pathways between Forskolin treatment and flow-derived cystogenesis. And so you can see these 6 compounds in compound 1 through 6. In the control, the CDH1 positive tubular structures are small. Under flow, you get cystic dilatation. In compounds 1, 2, and 3, resolve this cystic dilatation under flow, whereas 4 did so partially, and 5 and 6 were ineffective. The most effective compounds were largely result of RAC1 or FOS inhibition. And so next we look to do a deeper dive on RAC1 and FOS by first wanting to validate our findings in ARPKD samples. So we immunostained RAC1, or our collaborator immunostained RAC1 and FOS in cystic distal tubular epithelial cells and found that about nearly 100% of distal tubular cells were RAC1 positive compared to about 10% in controls. Similarly, 50% were FOS positive as opposed to about 15% in controls. So we were trying to validate our finding in organoids and prove that RAC1 and FOS was indeed upregulated under flow, and found that the same is true in ARPKD patients. So next we looked at differential gene expression between the double mutant underflow and the het underflow to look at transcriptional changes that were specific to the second gene mutation. And we really want to interrogate downstream mediators of RAC1 and FOS signaling. In normal cells, mechanical stress induces RAC1 FOS signaling axis off of RAC1 causes cytosolic remodeling. From downstream from FOS is negative cell regulator of cell proliferation. And so what we ended up seeing was that under flow in the double mutant compared to static conditions, there was a reduction of RAC1 mediated induction of these sort of cytoskeletal remodeling transcriptional signature. Whereas also under flow compared to static, even though FOS was induced, the downstream effectors were reduced across the board, suggesting that there's sort of upregulated RAC1 and FOS signaling, but that the downstream effectors are reduced rather than increased. The most interesting thing in my mind is that when we used RAC1 inhibitor and FOS inhibitor, it resolved the expression of cytoskeletal genes and these negative regulators of cellular proliferation, suggesting that it wasn't just decoupling, but we could inhibit them to rescue this transcriptional signature, which will be the basis of future studies. Thank you.
Thank you, Navin. Any questions can be put into the chat. We have time for some questions. I had one question about, so you're very interested in the effective flow rates. I was wondering how in your organ-on-a-chip system, the flow, have you tried different rates of flow, and does that make any difference in terms of the different pathways that you find, and how does it compare with what we might expect to see in vivo, in the in vivo situation?
Understood. Phenomenal questions. I would say in the original paper, when we did flow-induced sort of maturation and vascularization, we tested different fluidic shear stresses. Because this is sort of a closed system with very defined, sort of componentry, we can calculate the shear stress that is applied to organoids. It was about 0.2 dynes per centimeter squared. And so, at a high flow condition that induced maturation and vascularization. In a low flow sort of setting, we didn't see the same degree. So it seemed to be that there was, that the flow rate mattered. And that for this cystic, phenotype, we used the same high flow rate that induced maturation and vascularization. And so that was sort of fixed based on a previous study. And that flow rate, you have to keep in mind that these organoids are being superfused. They're subject to superfusate, meaning that it's not that flow is restricted to inside of vasculature and that the flow is really from, it's a luminal filtrate that gets absorbed into the paratubular capillary bed. But you can understand that, most of what the filtered load in the kidney is actually resorbed across the tubular epithelia. About 99% of your glomerular filtrate is reabsorbed. So this is a highly fluidic microenvironment. And our our rates of high flow are comparable to the interstitial sort of flow rate that is theorized, but we can't really directly measure that easily.
Okay, great. Thank you. I think in the interest of time, we should probably move on. There are a number of other questions in the chat, so I invite Dr. Gupta to respond in the chat to those as well.
Thank you.
Thank you again for a great talk. I think we'll move on to our second presentation. This is Yu Ishimoto, and he's affiliated with NIDDK in the Germino group, and he'll be talking about a novel organoid for ADPKD based on mouse nephron progenitor cells.
Yeah, thank you very much for your introduction. So can you hear me?
Yes.
Okay. So hi, my name is Yu Ishimoto. Today I'd like to talk about a novel organoid model for ADPKD based on mouse nephron progenitor cells. Several in vitro models have been developed to study ADPKD. However, cystic phenotype are not always genotype dependent, and widely used in vitro models for ADPKD research often based on not morphology but size of structure. In addition, need external stimuli like pulse calling to make cyst formation. More recently, some groups developed human iPS cell and ES cell-derived organoid system to evaluate PKD cystogenesis. However, these systems take long because there are no established method to maintain NPC derived from iPS cell or ES cell. And every time we have to differentiate them to make nephron progenitor at first. And also it will be difficult to do in vivo correlation. But mouse NPC, prior study established a protocol to maintain NPC using NPC reporter mouse. Collaborated with the authors, we started mouse nephron organoid system. Mouse NPC will be predicted for pure population than that of the others, and starting with pure population should reduce contamination of non-renal cells in nephron organoid. In addition, finding with mouse NPC will be easily validated with the mouse. This is an example of differentiation steps to make kidney lineage cells from iPS cell. We focus on NPC that can be differentiated into porocyte to distal tubules. We try to collect pure NPC from E13.5 mouse with antibody-based sorting that doesn't require reporter mouse. This is the approach we used. Robot2 is an NPC surface marker. NPC population includes somewhat differentiated to the next stage. And so to sort pure NPC population, we use podocarexin and PDGFRB combined with Robot2 and collected Robot2 highly positive and both podox and PDGFRB negative cell population. This is an example of a mouse NPC derived on nephron organoid. Staining with tubular markers, this is a bright field. As shown here, they differentiated very well. So next, I established NPC from PKD1 knockout mouse. And PKD1 knockout nephron organoid could make cystic structure spontaneously. These are the multiple replicate of single NPC line derived from wild type and PKD1 knockout mouse. This is the time course. PKD1 knockout organoid can make cysts at around day 10. We could also see cystic structure in other PKD1 knockout model. Here, I use PKD1 CKO tamoxifen clay mouse, and there are also multiple replicate of one cell line. These are the same cells. The upper one is before induction, and the lower one is after induction. This is the time course. They also make cystic structure at around day 10. This is an example of full-mount imaging of cystic nephron organoid with clarity. This is kind of the higher magnification. This is 3D projection of the same organoid. And as you can find, so preliminary data suggests primary cilia facing inside of the cyst, and it's different with that of the other PKD cystic organoid. This is the NPC line that I have established so far. I didn't evaluate all of them, whether they can make cysts or not. But so far, all the PKD1 knockout made cysts, and wild type didn't make cysts.
1 minute.
This is a conclusion. We established a novel mouse nephron organoid system. This is the first mouse nephron organoid model that can make cysts spontaneously. Our mouse nephron progenitor-based organoid system can be used more broadly to study other genetic kidney disease models. Thank you for your attention.
Thank you, Dr. Ishimoto. We're now open for questions. There was one question from Ronak Lakia asking, at what time point do you give tamoxifen in the in vitro inducible knockout system?
I induce the clean by using 4-hydroxytamoxifen, and it's maybe 2 or 4 days later, I can see deleted band of the PKD1, and that deletion, continue to very long. So that image that I show here is about several times I did the passage several times after that pre-induction.
Okay. Darren Wallace asks, did the cysts form within a collagen matrix or within the media?
That's a very good question. So I'm now try to find best condition to make cystic structure, and maybe it will be true. So using some matrix, it will be enhance cystic structure compared with the cysts like shown here.
Okay. And Brad Yoder asks, any idea why some organoids have cilia on the outside and yours are on the inside?
That's a very good question. So I'm not sure why it's something different, but cell is also different, and the differentiation method is also different. So maybe there are a lot of difference between compared with my organoid and other organoids. So maybe something happening, something happening there, and then my organoid can make cystic structure to go to the inside. But I need to do more to see using apical marker or basolateral marker to see whether cysts primary cilia facing inside of the cysts or not. Yeah.
Okay. And Paul Welling asks, how fast do the cysts form?
It's about 10 days. It takes about 10 days to make cystic structure.
Okay, and then I think a final question, Thomas Natoli asks, do you have thoughts on why the NPC cultures form cysts spontaneously while embryonic kidney organoids from mice require induction?
Sorry, one more, please. So please ask. Yeah.
Why the NPC cultures form cysts spontaneously while embryonic kidney organoids from mice require induction?
I think so. Embryonic kidney no need to require, especially when we use the PKD1 knockout embryonic kidney.
Okay. All right. I think in the interest of time, we'll move on. Thank you.
Thank you very much.
For a very nice talk. Any additional questions can be posed and answered in the chat. Our third speaker for this session will be Thomas Naert, who is from the Institute of Anatomy at the University of Zurich in Switzerland. And his presentation is titled Xenopus Animal Models for Autosomal Polycystic Kidney Disease with Automated Deep Learning Analysis of Cystogenesis.
Thank you for the introduction. So indeed, today I would like to present our novel animal models in Xenopus for autosomal polycystic kidney disease. So Xenopus, a frog and an amphibian, features external embryonic development, much akin to zebrafish. And this is great because it really allows us to deliver macromolecules to the developing embryo. So for instance, here one can in fact see a fluorescent dextrane, of which the expression is specific to one side of the developing embryo. Another great thing about Xenopus embryos is that already at 2.5 days of age, they have a fully functioning kidney. In fact, this tangle that one can see here in this immunostain, it is, you would think it's a glomerulus if you're used to looking at mammalians, but this is in fact a pronephros, a fully functioning kidney, which is built up out of cells that share characteristics with proximal tubule, intermediate tubule, and etc., compared to mammalian systems. So we wondered what happens if we do PKD CRISPR-Cas9 and we restrict this to one side of the developing embryo, thus performing gene editing and PKD1 on the left side of the embryo. And in fact, already at 2.5 days of age, we get an in vivo model for cystogenesis within Xenopus. So we thought this was super cool, because we can inject so many embryos at once, we really wanted to go to a more high throughput approach. This thinks about chemical screens, thinking about CRISPR screens. So for this, we had to set up an automated imaging analysis pipeline. So we had a large data set consisting out of wild type Xenopus, showing this nice little tangle of pronephros again, and several guide RNAs eliciting cystogenesis within Xenopus. So we trained a first neural network called 2D Nephronet to basically isolate the kidney from these images. And this allowed us to then crop it out. So at that point, we could train a classification, artificial intelligence, a classification deep learning network. So you might know that deep learning networks or AIs are able to, for instance, if you give them a picture, say, is this a dog, is this a cat? So you can do similar things with Xenopus pronephros. You can train an AI to recognize either a normal kidney or a cystic kidney. So for instance, here, the normal kidney has a lot of firing in the normal channel, while this very cystic kidney has almost a dramatic firing in the cystic channel. And a nice thing is that these deep learning networks, in fact, interpolate between these several pathological states, and thus having these large image. So if having a lot of these images from the tadpoles, we can do fully automated image processing, getting the kidney area from the first deep neural network, but then also getting a measure of cystogenesis of what the percentage of the area of the kidney that is cystic is. And this in a fully automated manner, showing that cystic kidneys only occur within the injections of PKD1. We also questioned whether we could do something similar with PKD2. So we injected once again PKD2 CRISPR-Cas9 doing gene editing, and we restricted this to the left side of the tadpole once again. So what we basically saw is that we could see cystogenesis occurring very pronouncedly again. But the nice thing is, because we delivered this gene editing specifically to one side of the animal, so across the lateral midline, we see the development of a normal kidney on one side, here on the right, and a cystic kidney on the left side. And this is really great because, in fact, you have an internal control within the animal. Having said that, from this large data set, which easily encompass a gigabyte from the light sheets, we also wanted to do 3D reconstructions. So for this, again, we trained deep learning networks for 3D reconstruction of kidneys from these larger Xenopus tadpoles, and this holds across the phenotypic spectrum. And then finally, this also allows us to do feature extraction approaches. So for instance, here we use automated image analysis to look at the diameter of this of the kidney tubule as it progresses through the pronephros. And indeed, we are able to show that upon local cystogenesis, these heat maps are in fact firing, showing that there is a distension in this tubule, and this can all be done in a fully automated manner. So with that, I would like to thank the funding sources for this work, and would like to state that this work was recently published in Development, and that we are looking forward to potential collaborators in the field. And all the people that were involved in the work, which evidently I don't have time to acknowledge all of them right now, and you for your attention. Thank you.
Thank you very much, Dr. Naert. Any questions in the chat? I'll start out with, I was curious, so this is a model in frog or Xenopus. So to what extent do you think it recapitulates mammalian ADPKD models? Is there any benchmarks that you can use to compare? And also, can some of these deep learning tools be somehow applied to more advanced models like mammalian systems, like mice or even humans?
So to answer your first part of your question, so recent single-cell RNA sequencing studies really showed that large part of the Xenopus pronephros have very, very similar transcriptional programs compared to mammalian kidneys. There are, of course, some differences, such as the loop of Henle not being present. However, we feel that as the pathogenesis of ADPKD really mostly occurs within the tubules, and part is very well conserved across the species that we study, that this in fact does hold. To answer the second part of your question, yes, I do think that these deep learning analysis methods can be used in more mammalian models, definitely. Also part of what we present in this study is in fact the tools to allow researchers to use these deep learning approaches in their own research without actually having to have a master in computer science, because it's all Fiji plugins that we're trying to relay to communities that they can have access to these very powerful tools.
Great, thank you. I think in the interest of time, if there are any other questions, they can be posed and answered in the chat. We'll move on to our fourth speaker of this session, and that is Nikonorova from Rutgers in New Jersey. And she will be presenting a brief talk on the identification of novel polycystin-associated cargo of extracellular vesicles in C. elegans.
Thank you so much. You can hear me and see the slides, right?
Yes.
Human polycystins function in the cilium and extracellular vesicles. And we know that urinary EVs from patients with ADPKD differ in their composition from unaffected patients. And they also promote cystogenesis in 3D cultures. Polycystins are incredibly conserved, and we use the roundworm C. elegans, where it's also function in cilium and vesicles, to answer fundamental questions. And today I'm going to talk about the question that we pursued recently: what proteins colocalize with polycystins and those vesicles? Because their identification of those novel proteins might produce better markers for ADPKD. And in C. elegans, polycystins are expressed in cells that release vesicles into the environment. And recently we identified that overall they secrete about 3,000 proteins into the environment from the polycystin cells and all other tissues. So to increase our specificity of identification toward polycystin vesicles, we introduced biotin ligase, proximity labeling enzyme, into polycystin vesicles. So this turbo ID would be targeted to polycystin 2, and that would label all the environment around it. So our mass spectrometry after this experiment came back with less than 15 candidates. And today I'm going to talk about, I'm going to show just 8 that we were able to validate as a true cargo and true interactors of polycystin 2 that go to the same vesicles from the polycystin expressing cells. And beside this PC1, worm PC1 that's called LOV1 with the gain and GPS domain, we discovered a number of adhesion molecules, and one of them 7 transmembrane protein that reminds GPCRs, but it lacks actual GPS and gain domain, and a very similar protein that also has lectin and EGF domain, but the first 2 alpha helices were disrupted in evolution by introduction of a negative amino acid, single pass lectins, and some transmembrane proteins that vaguely remind sodium channels of bacterium. And it has a very conserved core with many orthologs in human, including human traps, and then C. elegans, those are TRF1, TRF2. Some zinc finger protein that is related to human zinc finger protein, and we know that this in mice, for example, causes a retinal malformation and skin lesions. And also we discovered nuclear shuttle, which is human NTF2-like protein. And for the sake of time, I just want to focus on just one candidate, let's say TRF1. So I'm going to show you how we work with this. We use genetic tools to determine how these signaling complexes are assembled within the cilia and within vesicles around polycystins. And so just I'm showing you right now, this is the male tail. And those elongated structures that you see, those are rays, and they mechanosensory organs that the male uses to find the hermaphrodite, defined mating partner. And they all labeled numerically, and you can see the ray 4 at the very end of the dendrite of the neurons that express polycystin. You can see cilium transition zone excludes TRF1 in this particular example. And you can see TRF1 within the cilium and the very, very tip. And you can see, for example, in ray 9, over here, TRF1 is released in vesicles into the environment. You can see it easily on the microscope. And then in ray 3 is also in the process of abundant EV release. And now we use genetic tools available in C. elegans. We look at the polycystin-2 mutant. You can see again presence of TRF1 in the cilia, but there is no presence in the very tip, and there is no vesicles released. So the similar results, and that's true for all the rays and for all PC expressing cells. From this, we can make a conclusion that polycystins are required for cargo loading to ciliary vesicles.
1 minute.
And we also check that for a number of other cargos. That is also true for other cargos. Yeah, now we think that we're figuring out the hierarchy of assembly of these complexes within the vesicles, and we think that many molecular interactions that we discover in worm might be also realized in human polycystins as well. Thank you.
Okay, great. Thank you very much, Dr. Nikonorova. So we have time for a couple questions. Perhaps I can start off. I was curious about the presence of PC1 in the EVs that you're getting. Do you see that? Are they associating with the PC2 that is expressed? And have you tried also knocking out PC1 and seeing what the effects are?
Yeah, for this particular cargo, PC1 knockout gives a similar phenotype. So there is no enrichment at the very tip of the cilium. The soluble protein is not delivered there. Yeah, and there is no vesicles seen. And the PC1 affects number of other cargos, but not all of them.
Okay, great. Thank you. Gabrielle Loeb asks, what is the zinc finger protein that interacts with PC2?
So it's a ortholog of human ZC2CH1A. I mean, in worm, it doesn't have a name yet. It just has a cosmid number identified when genome was sequenced. We're going to name it at some point. Okay,
great. If there aren't any other questions, I think we'll move on, and thank you for a very nice talk. Our last talk in this session is given by Cynthia Sieben, who is in the Department of Medicine, Division of Nephrology and Hypertension Research at the Mayo Clinic in Rochester, Minnesota. And her talk will be Comparative PKD1 and PKD2 Missense variant profiling aids molecular diagnoses across the ADPKD spectrum and reveals common pathomechanisms.
Thank you for the kind introduction, and thank you to the organizers for giving me the opportunity to speak here today. The project aim is to develop improved methods to determine PKD1 and PKD2 missense variant pathogenicity. And here, pathogenicity of 48 PKD1 and 44 PKD2 missense variants was determined using multiple methods. First, we used two different guideline models, the American College of Medical Genetics and Genomics, or ACMG, as well as variant strength group categorization. The ACMG is the current clinical standard, and the VSG scoring is our current research standard that we developed in our laboratory. And these two are similar in that they incorporate data from multiple sources, including patient and population data, computational data, and data from other sources. However, the emphasis and weighting of individual components is different, and ACMG allows for inclusion of functional data where available. Second, we use two different in silico scoring methods that have been developed more recently, Revel and CAD. And both of these tools incorporate predictions from multiple computational tools and use machine learning to predict variant pathogenicity. Lastly, we developed a 3-color flow cytometry in vitro assay. And as depicted below, We use a kidney epithelial cell line that's isolated from cyst lining of an ADPKD patient. We then exogenously co-express mCherry PKD1 and GFP PKD2 and label with an mCherry antibody and a Lexaflor 647 secondary antibody to detect surface polycystin-1. The cells are then analyzed with flow cytometry, and we use polycystin-1 localization to the apical plasma membrane as a readout. So here I'm showing a summary of our comparative results for monoallelic PKD1 and PKD2 variants that we've assessed. We've also evaluated variants of complex inheritance, but I'm just showing the monoallelic here for simplicity. To clearly represent the results from these 5 methods, we have pseudo-colored heat maps. And the key to the right here shows the pathogenicity categories that we're using. Variants that clearly impact splicing are also noted, although a full assessment still needs to be performed. As you can see from the heat maps and the summaries to the left, using the ACMG guidelines, very low levels of pathogenicity are predicted for these variants, whereas all of the other 4 methods predict a larger number of variants to be pathogenic. Comparing the 2 guideline models, ACMG and VSG, you can see that our VSG categorization predicts a much larger number of variants to be pathogenic, likely due to differences in category weighting. And I would also note that our VSG method has been designed specifically for PKD1 and PKD2 variants. Using our VSG categorization as a standard, both of these in silico methods, Revel and CAD, also find a large number of variants to be pathogenic. However, Revel seems to be less effective for PKD1, and both of these find some of these predicted neutral variants to be pathogenic. Our in vitro assay also shows a large number of variants to be pathogenic. This is the last column on the right. And the difference in pathogenicity categories here indicate differences in allele penetrance, and the cutoffs and breakdowns that we use are shown here to the right. Some of the variants that are predicted to be pathogenic using the other methods are found to be neutral based on the in vitro assay. These shown here in blue, which may indicate increased sensitivity of the assay or that other pathomechanisms are impacted. However, as you can see, a few of these also impact splicing, which is not assessed with our in vitro assay.
1 minute.
Thank you. Using correlation analyses to assess the consistency between the latter 4 methods, we find that our VSG categorization and the in vitro assay are the most consistent, which is shown here in these plots for PKD1 and PKD2. And as a note, our VSG categories are classified between 1 and 6. And here I have included all of the variants that we have assessed, including the biallelic and digenic variants. A key showing how we break up these categories is listed here, as well as the breakdown for the in vitro assay. And I would note that our in vitro categorization and in vitro assay are also the most informative, as both of these provide information on allele penetrance, and the in vitro assay also provides some information on pathomechanisms. Revel also showed good consistency with our VSG categorization over CAD. And in conclusion, these studies highlight the need for improved methods for determining variant pathogenicity. And we find that the use of a guideline model is most reliable, as single bioinformatic tools are less efficient. And ultimately, incorporation of additional tools and information into the guidelines, such as the in vitro assay or adjusting the weighting for individual components for ACMG, will provide a more efficient and reliable method for predicting the pathogenicity of non-truncating variants. And although this was not the main focus, today, these studies also highlight that defective PC1 maturation and trafficking to the plasma membrane is also a common pathomechanism in ADPKD. And with that, I would like to thank all of the co-authors for their contributions to this work and our funding sources for their support. And thank you all for your attention, and I would be happy to take any questions.
Thank you so much for a nice talk. We're open. We have time for a couple questions. maybe I could start. So all of these mutant variants that you tested are known to be pathogenic in patients. Is that correct? They're the single mutation identified, or is it possible that some of them are not non-pathogenic? In other words, is the gold standard, should it be 100% predicting pathogenicity?
So these variants are all found in ADPKD patients in a monoallelic inheritance pattern. However, we don't necessarily know that all of them are disease causing. We would predict that these 5 that are scored as neutral are likely not disease causing variants.
Okay. So so you're really just trying to find the best system for prediction. And at present, there's no single gold standard. Maybe a combination of some of them would be
Right.
predictive.
Our VSG categorization method seems to be fairly effective, but it's a bit complicated. But the ACMG guidelines is also a bit complicated. But as you can see, the ACMG guidelines are much more stringent since it's a clinical prediction.
Okay. There's a question in the chat. What cell model are you using for your in vitro assay?
So the cells that we're using actually come from that RCTE 97912 series. So we use the 9-7 cells and use CRISPR-Cas9 to delete or disrupt the remaining allele of PKD1 and both alleles of PKD2. So it's a kidney epithelial cell line.
Okay, great. Thank you. All right. If there aren't any other questions, I wanted to take this moment to thank all of our speakers and the audience participants for a great series of talks, very exciting new developments in the field. And I also wanted to make an announcement that all of the abstracts for this, in addition to additional abstracts, can be found on the symposium websites. And posters can be found on the PKD RRC website under the Educational Resources tab. The files are very big and will take some time to download, but that's where everyone can get more information.